As I was introduced, I'm Jason McDermott. I'm a senior research scientist at Pacific Northwest National Laboratory. For all of those who don't know, that's a Department of Energy facility from the U.S. government. We are essentially run by a research institute, and we're located in the eastern side of Washington State. So Seattle has a reputation of being very, very rainy. We're actually almost in a desert on just 200 miles east. So I'd like to talk to you about this paper that I published in April in PLOS Pathogens. It describes a machine learning method that we developed to identify type 3 secreted virulence effectors from gram-negative bacterial pathogens, specifically Salmonella Typhimurium. So I'm going to give you a little overview of the results of the paper and then kind of walk you through some of the implications that the results brought and some of the structural follow-up that we've been following up on a couple of these hypotheses. Briefly mention some of the drug discovery options that are available for the future, and finally wrap up with how this ties in with kind of my group's overall goal, which is modeling host pathogen interactions on a systems level. So I mentioned a focus on Salmonella Typhimurium. That's pretty much the organism that we're focusing on for the systems biology models. Salmonella Typhimurium is a gram-negative bacterial pathogen. Its normal route of introduction into the host is orally. It can invade through the intestinal epithelium under certain circumstances, enter the bloodstream, and it's actually taken up by macrophages whose job it is to go around the body and clean up these kinds of bad things happening. Salmonella has a couple of dirty tricks up its sleeve. It activates a very complicated virulence program upon entry into the phagosome, and it actually rearranges the host cell network and creates its own intracellular niche into a salmonella-containing vacuole. So it does this primarily through the use of virulence effectors, which it injects directly into the host cell cytoplasm through these type 3 secretion systems. Salmonella Typhimurium has a very broad host range. It actually kills nearly 1 million people a year, some number like that, mainly in developing countries. If you're from the U.S., you probably know in the last couple of years, Salmonella has been responsible for a couple of foodborne outbreaks. One, I think, was in salsa or tomatoes from salsa, and the other was in peanut butter. Importantly, Salmonella Typhimurium is a nice model system for Salmonella typhi, which is the causative agent of typhoid fever, and that has a very narrow host range of humans, so it's a lot harder to investigate. On the right-hand side, this is a schematic diagram. It's very complicated. You don't have to worry about it too much. Basically, the pathogen program is on the left, and the host program is on the right. This is my actually fairly simplistic idea of what's going on. Salmonella Typhimurium has 2 pathogenicity islands. These are genetically distinct regions that encode 2 separate type 3 secretion systems. Again, the type 3 secretion system's job is to inject these effector proteins into the host cell where they can go and muck with the host cell network. For Salmonella Typhimurium, there are about 37 known secreted effectors, which I'll talk about in a minute. So, I'm a Mac person. I keep on pressing the wrong mouse button here. So a little overview of the type 3 secretion system. There's a schematic diagram in the lower left. This was courtesy of our collaborator, Fred Heffron at Oregon Health Sciences University. It's really an elegant molecular machine that spans both the inner and the outer membrane of the gram-negative bacteria. It forms a needle-like structure, actually sometimes called the injectosome, which can penetrate into the host cell membrane, and it's a very narrow pore. It specifically injects proteins into the host cell cytoplasm. So these collection of secreted effectors are specifically targeted under the right conditions to this type 3 secretion system. So some of the problems and what really drove this work is, I thought was actually kind of nicely introduced by Danny, talking about remote homology. So these, in a way, this is a problem of remote homology. We know that all of these effectors have the same function in one way. They all are targeted toward the type 3 secretion system. We also know from many experimental studies in Salmonella and elsewhere that if you cut off the N-terminal region up to about 30 amino acids, you lose this function. So this N-terminal region is very important for function. However, there's no conserved sequence motif that directs these effector domains to the secretion system, unlike in some other secretion systems where you can actually identify these by bioinformatic methods. This doesn't seem to be possible, at least up until the point that we came in and solved everybody's problems. So there's diverse sequences. They're all coding for a common function. Although there are a number of known structures of type 3 secreted effectors from a number of different organisms, in all but one case, the N-terminal region is absent from electron density and crystal structure, or it has to be cut off before these things will actually crystallize. So the hypothesis that has kind of emerged because of this information and because there is really no sequence similarity between these secretion signals is that we actually have a disordered sequence. But this is kind of unsatisfying in a way. How is this, how are these different sequences actually coding for the same function? So really you can kind of explain this by two hypotheses. The first is kind of a ridiculous one, and that is that for every different effector and every different secretion signal, there's a different mechanism for targeting. This seems ridiculous on the face of it. There's 40 different effectors and secretion signals in Salmonella Typhimurium. There's actually some evidence for this because some of these effectors actually have cognate chaperones. So these are proteins whose specific and sole job, apparently, is to chaperone one effector to this type 3 secretion system. This has to do with the unfolding of the effectors that has to occur to thread through this narrow pore. And there are actually a number of examples where there's no chaperone and these things are still secreted, and there's some experimental evidence that you can actually delete the chaperone in some cases and still get secretion. So the second hypothesis, and the one that I'll talk about more today, is that maybe these different sequences are all coding for a similar structure or a similar set of structures. So I'll talk about that in just a minute on the next slide, or in a couple of slides. But I want to give a little background on what the state of the art for detecting novel secreted effectors in novel pathogens was before this. There have been several bioinformatic techniques. Most people used a simple blast search to find things that were similar to their effector domains. So this is not the secretion region, but the region downstream that actually does the work. There's actually quite a bit of, there's a number of families of these. This works very well for places where we know secreted effectors and we know the effector domain. If you do a survey of recent literature, you find that these new effectors are being discovered on a fairly regular basis, and a lot of them don't have any sequence similarity to other known effectors. So this is kind of a limited way of looking at identifying secreted effectors. So there's also a couple other bioinformatic methods. I won't really go into those details right now, but mainly they were focused on single organisms, and they seem to work okay for those organisms, identifying some secreted effectors from those organisms, but they weren't really extensible to other organisms. So kind of driving our project was this idea that it would be very nice to be able to characterize the virulence effector repertoire of known or novel pathogens. We developed a machine learning approach. This is actually a very simple in terms of machine learning. We named it the SVM-based identification and evaluation of virulence effectors, or the SIEVE method. Every machine learning method, as we all know, has to have a cute acronym. This is ours. And we used a number of different features that were derived from primary sequence. Some of these have been used fairly commonly in these kinds of approaches. We used amino acid composition. We used a couple measures of evolutionary conservation for the whole domain. The amino acid composition in the N-terminal secretion region. We used the GC content of the underlying gene. We used a phylogenetic profile that we assembled from a number of different organisms. And finally, importantly, that I'll talk about in a minute, we encoded all the N-terminal 30 amino acids and just input these in an unaligned fashion. This was our naive approach at first. It turned out that it worked very well. We focused on two organisms for the in terms of the paper. Salmonella Typhimurium, which I've mentioned, has a fairly well-characterized repertoire of secreted effectors, 37 plus or minus. Pseudomonas syringae is a distantly related plant pathogen. It's also a gram-negative bacteria, has type 3 secretion system. It has a very nice system for identifying secreted effectors, so it also has a very large repertoire. The strain that we used, we had about 35 known secreted effectors. We used those as the positive examples for our training. For our negative examples, we used everything else in each genome. Now, this runs into the problem that one of the things that we're trying to do is we're actually trying to identify new secreted effectors from the genome of Salmonella Typhimurium. So we know that there are probably some examples that are in our negative examples that we just don't know about. It turns out that this seems to work okay, and we're able to identify some of those, and I'll talk about that in a minute. This is the results that we reported in our paper. This is a specificity sensitivity curves for the two models. So one trained on Pseudomonas syringae effectors and tested on Salmonella Typhimurium, and the other trained on Salmonella Typhimurium and tested on Pseudomonas syringae. We also did a leave one out analysis of cross-validation of the Salmonella Typhimurium. We got very similar results. The bottom line here is that our ROC area under the curves are 0.95 and 0.96. This is very good. It's much, much better than any of the other methods that had previously existed, including just identification of blast effector domains that were similar. I should note that we also first went through these effector libraries that we had, and we eliminated the places where we could actually detect sequence similarity between the effector domains. So we kind of tried to have as pure an approach as we could. So this allows us to identify novel secreted effectors across organisms. We actually applied this to a third organism, Chlamydia trachomatis, which is a largely genetically intractable pathogen. Because it's largely genetically intractable, the predictions we made we haven't validated, but we did make some predictions in Salmonella Typhimurium, and several of these have been validated either by our group or by other publications that came out about the same time. I listed a few of the other predictions here, not necessarily the highest scoring predictions. Confidence here is the chance that you're getting a false positive, essentially. So if you 50% confidence means that you can have two predictions and one of them will be right. And these these three I picked out because they turns out that they've had structures solved by structural genomics projects. We're actually working with the structural genomics project centers that are established by the NIAID to provide better targets. But this is kind of interesting, because it shows us that maybe we have some drug discovery targets that are sitting right here, and we didn't really know about them. So that's kind of the first thing we can do. And I'll talk about the second thing we did in the paper in a minute, but just, no research is ever done in a vacuum, and we'd been working on this for 4 years, I think, since we'd actually come up with the idea and run through a number of different iterations and talked to our biological collaborator quite a bit. We published in April in PLOS Pathogens. In the same issue, there was a second paper that was using very, very similar approaches as us, coming to very similar conclusions as us. There's a couple of differences that are pretty interesting that we're trying to suss out right now. There's also an opinion article that was written on both of these. And then more surprisingly, last month, there's a third paper. So they, again, used a machine learning method to identify type 3 secreted effectors. I have a student right now who is working on evaluating the similarities and differences between all three of these different methods because they have some subtle but important differences in the kinds of training sets they were using and the kinds of features that they were using. So this is very heartening because it says that other people in this area are also looking at the same problem and coming up with the same kinds of solutions. It was also fairly frightening because we could have been scooped at any time during that, especially up in the last year. I'm not sure why we weren't, but I guess luck. So the second thing that we did with this, we have all this sequence information that is in our model, and it is important for classification. We wanted to see what sequence features were actually most important for classification in both of these models. And so we used a method called recursive feature elimination. It basically allows you to successively remove the features that are less important. So you minimize the number of features that give you the maximal performance. We got some interesting results on the phylogenetic profile. The GC content and evolutionary conservation were both very important. But we also got a sequence pattern, and these are the most significant sequence biases in both of these models that still allow accurate classification. If you actually just use sequence, you still get a fairly good classification as well, so if you ignore the rest of the features. So this is for the Salmonella Typhimurium tested model, and this is for the Pseudomonas syringae tested model. The gray bars are places where you get agreement with these. I should note, I say motif here, but this is in no way the kind of conserved motif that you would think about in terms of, none of the effector secretion signals actually look like these consensus signals. We're actually trying to use this to test whether or not we can create a consensus signal out of this that will actually be secreted. That's work in progress through our collaborator, Fred Heffron, right now. So suffice it to say, this is very interesting because we actually, in the paper, we describe how it agrees with a number of experimental results that have been published, as well as the other bioinformatic models that have been developed for Pseudomonas syringae is the key example. It agrees with that very well, but the model that they, or the patterns that they were finding don't map across the organisms very well. So that's kind of where our paper left off. And now I'll talk about some other results. But very briefly, we have this, all the sequence information in our model. And so we thought, well, this would be an interesting thing to do. We could use an in silico mutagenesis method. This is basically analogous to an alanine scanning mutagenesis that you would do in real bench science. In this case, we mutated to every other residue, and then we see where those mutations have the biggest impact on our ability to classify the individual proteins. Unfortunately for Salmonella Typhimurium, there are pretty much only 2 examples where they've actually done experimental work that would help us validate this approach, and these are marked in kind of gray-pink here, where they've identified these important regions. And we see that we get a peak in our model, but It's a little bit unsatisfying at this point. Dana Sharp, who's a summer student, I'm having her go through the literature and find all the places where they've done mutations in type 3 secretion signals from different organisms. And we're compiling this into a very nice list of positive and negative secreted examples, because these are ones where they've actually tested and said this protein is not secreted. So we're hoping that that actually gives us a very nice model that we can extend this idea even further. So I want to talk next about this hypothesis that maybe these different sequences are actually giving rise to some kind of similar structure. We looked at this using a number of different structural prediction methods. We first used CyPred to predict secondary structure of all Salmonella proteins. We found significantly enriched region of beta sheet in the N-terminal region. This is significantly enriched coil, which is not very informative. We also looked at disorder prediction and found that these two regions were more likely to be disordered in these secreted effectors. This region seemed to be ordered in that there were fewer occurrences of disorder prediction. We then used a tertiary structure prediction. We used actually a set of 3 different related but different tertiary structure prediction methods to predict 20 structures for each of the 30-some secretion signals. We then did structural clustering, so looking at minimizing the RMSD between all of these structures, and we found a very nice cluster. And this is kind of shown here in overlap, but you can actually see it better here. So this is really what the consensus structure looks like, is in color here. This beta turn, exactly like we predicted in our secondary structure, followed by an alpha helix. And this is where it gets kind of exciting, because the only known structure of a type 3, N-terminal type 3 secreted effector is in YOP-H, and that's shown in gray here as a very nice alignment. So we think this really might say that, this seems to suggest that these different sequences are capable of forming the same kinds of structures. We also did some molecular dynamics simulations. I'll go over this really quickly. Residue number is on the y-axis. Time of simulation is on the x-axis. The color indicates formation of secondary structure, red being beta sheet. So we see that same beta sheet being formed. This is just one example. We don't see a corresponding alpha helix. This is work being done by Roberto Linz at Pacific Northwest National Lab. And we're trying to extend this to some more examples. So this has all been predictive so far. We had 4 of these peptides, the N-terminal 30 region, synthesized. This is the example from one of these where we've done circular dichroism. This is circular dichroism in just the buffer that is used at and you see that there's actually not much secondary structure here, so these might actually be flexible, disordered in solution. As you add more and more TFE, trifluoroethanol, this is a solvent that's commonly used to induce structure in partially ordered peptides. You see this formation of order. This is very suggestive that under the right circumstances, this can adopt some kind of ordered structure. We've actually got NMR results that are being processed right now. This is very early. Gary Buchko at PNNL is working on solving this structure, and he says that it shouldn't be, probably in a matter of a couple of weeks, we'll have a structure for this. So I've kind of outlined how we've identified common sequence elements and common structural elements, or at least predicted structural elements. We're trying to follow this up with some experimentation. We're very hopeful that we can actually find some kind of common structure. What we really think might be happening is that these secretion signals are partially disordered in solution. They may be adopting a set of low energy states. And one of the things that we're trying to do is on some of the other more problematic NMR, we're trying to combine that with the molecular dynamics simulations to get a view into what those different states are doing. What are the conformations of those different low energy states? And this may also be very important for binding to the secretion system apparatus. So these may be disordered in solution, and they may adopt a fold upon binding. So this also makes a very nice target for drug studies provided that we know what these actually interact with in the type 3 secretion system, which currently we don't, but that's another avenue of investigation. So I'd like to just briefly go over a systems biology project. We've used a number of different transcriptomic and proteomic data sets to derive transcriptional regulatory interaction networks, functional interaction networks from Salmonella Typhimurium data. We've actually used some topology to infer very important nodes in these networks as bottlenecks. This is similar to other work that's been done in the yeast protein-protein interaction network. And really what we're thinking is that this ties very nicely into with our secreted effector prediction, because if you think about where the secreted effectors lie, they are at the interface between the host and the pathogen. So if we really want to model the two networks working together, we have to know all the points of intersection. And so what we're hoping is that this method will allow us to more completely model the host-pathogen interaction as we go along. So just to summarize, we've developed a method to identify type 3 secreted effectors. We've identified some commonalities in sequence and structure between these. We have some experimental data showing that our structural hypothesis, these different divergent sequences might be similar, might be correct. We're working on extending it to some other systems, and we have a prediction server that we're going to roll out later this month. I'd just like to thank everybody. I mentioned most of these people. Fred Heffron is at OHSU. He's really the Salmonella expert. Ram Samudrala, my former postdoc mentor, has helped me out with a lot of the modeling. Funding sources. Shameless plug here for postdoc. We're hiring. I've got flyers out in the ISCB board. This is for the systems biology of host pathogen interactions. And finally, one more plug. I have the Art and Science exhibit on a completely different subject. So thank you.
Thank you very much. We are open for some questions. Maybe I should start. Did you mentioned this idea of mutagenesis and some kind of alanine scan. Did you thought of trying to do the best adapter in the world? I mean, to take and make something that will work more than our natural?
Yeah, we've got a couple of things going right now. So one of the things is this consensus signal. So it's not clear that a consensus signal would be better.
I don't think so.
But the other thing that we did was actually we did scrambled sequences. So it's interesting, these fall out into two groups. If you randomize the sequences in the N-terminal regions, some of the effectors the wild type is always best. Other effectors, you find that you get a distribution. You get some that are much better predicted than the wild type. So we've actually got two of these. We took the SSEJ that we're characterizing right now. We're having peptides made of one of these that's way, way better than the wild type and one that's way worse. And we're actually going to have those put into mutants. And we're hoping that we can actually get better secretion with our using our model.
We can use some energetic design approach. Please.
Peter Karkas, CRI. You mentioned in other organisms some motifs had been identified for type 3 secretion systems. Does the RXLR motif ring a bell? I think it's Erwinia family.
Yeah. So what was the question? What was the question there?
You mentioned in other species motifs have been found by more straightforward approaches.
Yeah.
And
I'm very familiar with the Pseudomonas syringae work. Mm-hmm. I'm not so familiar with the. You were saying RXRL?
Yeah, it's an R than any amino acid, then a leucine and an R.
And that's for type 3 secretion?
I believe so, from memory. I was trying to check the reference, but the wireless is down.
Yeah, I'd be interested in seeing that reference. That sounds familiar, and I think I probably looked at that at some point. What we found with the Pseudomonas syringae, it was kind of, they had some prosite kind of patterns, so string matching patterns. and a series of these that they used. And when we applied those as they published it and tried to get as best results we could on Salmonella Typhimurium and a couple other pathogens, we didn't really get very many results.
There's another interesting parallel there with the malaria parasite, which was interesting.
Yeah.
Thank you. So I think we'll conclude this highlight session, and thank you very much to speakers and to you audience.